Okay, starters. So simple classical model. What we've got here is a stretch of DNA, and this box symbolizes a sequence of DNA coding for one gene. And next, right next to it, once it finishes one of those stop codon, stop triplet signals, right after that comes the stretch of DNA coding for the next gene and the next gene. And this is what DNA is about. It's the sequence of genes there. And what you would obviously then get is this arrow having this intervening step of that RNA stuff, but eventually producing an amino acid that produces a protein of this shape, and this is the protein coded for by this gene, this, and so on, and that's exactly how it works. That's the structure of DNA. Then, though, people began to find that that's not the structure of DNA. And what you began to get instead was something vastly more interesting, which is that for starters, when you look at coding for one single gene, it's not necessarily coded for in one continuous stretch of DNA. In other words, it's broken into little pieces, and you will have a stretch of DNA coding for the first third of the protein, and then a bunch of DNA that's got nothing to do with it. Stay tuned. Then coding for the next third, coding for the next third, that the gene was broken up into separate coding domains. And the term that was given for this was, these were called exons, and the in-between boring stuff were called introns. And this was a major finding, which made no sense whatsoever, because how are you going to get from this to then having a protein which has its normal sequence and shape, where this part was coded for by this exon, exon 1 of this gene, and this from exon 2, this from exon 3. That's a completely different sort of world of stuff. How are you going to do that? Because once you're going to make an RNA that's going to encompass all of this, and that's going to code for something completely different, because you got these introns in between there, and people then guessed something had to exist, which was soon discovered, enzymes called splicing enzymes. And what they did was exactly what you need to do to solve this, which is at the RNA level, these splicing enzymes would come along and they would snip out the part corresponding to here, and another would snip out there, and another one, as an enzyme catalyzing, would stick these two pieces together, and thus you had this utterly bizarre world in which genes, the vast majority of them, are not coded for in a continuous stretch of DNA, but instead are broken up into these separate exons, and then you need these splicing enzymes to clip out the boring in-between parts, the introns, stick them all together, and you've got your functional gene. Weird. Okay, but that's how they work, though. And it was shortly after these were discovered that one of the giants in this business, a guy named David Baltimore, who got the Nobel Prize for some of the work on that reverse process of RNA viruses turning back into DNA, he was the first to really appreciate that what you got here is a potential for a lot of information. Because of, and I think he was the first person to introduce this word into thinking about it, because of the modular construction of genes. Because genes come in these separate exons. What does that begin to allow you to do? Very important stuff. Okay, so we have the same structure here, and we have a gene coded for in a modular way in 3 separate exons, and thanks to splicing enzymes, protein catalysts, clipping this out, you wind up with this. What Baltimore was the first, or one of the first, to appreciate was that you could wind up with something different there as well. You could, for example, create a very different protein, one consisting only of A and B, or one consisting only of A and C, or one consisting of B and C, or A alone, and suddenly you have this combinatorial possibility of cranking out a number of different ways of putting together 7 different ways, since you can't transcribe this gene so that there's no transcription, 7 different ways of combining these different exons, 7 different types of proteins you could generate from the same gene. This was not accepted with a whole lot of pleasure by the old guard, because intrinsic in the know the DNA sequence specifies amino acid, protein shape, protein function, intrinsic in that is one gene specifies one protein. One gene only specifies a single protein. One protein is only specified, coded for by one gene. And suddenly this modular business allows you, with one gene, to generate 7 different kinds of proteins. For starters, how could that possibly work that way? Just on a nuts and bolts level, all you need are splicing enzymes that work a little bit differently in different parts of the body. One that will splice this off and is attached to an enzyme that will degrade this, while the splicing enzyme here does. And what have you just produced? You'll get this one. Coupling of splicing enzymes with degradative enzymes, and suddenly you've got a means to have tissue-specific expression of genes. The same gene will produce different types of proteins in different parts of the body because of splicing enzymes working differently. Then, just to confuse things even more, people began to note that there would be some splicing enzymes and genes where they would splice at a different point. And you would now have a gene with A and A prime and other splicing enzymes that would cut at other points. One single sequence of DNA generating all sorts of different types of proteins in different parts of the body, at different times of life, under different circumstances, in different individuals in different ways. Suddenly, there's a lot more information floating around in there. So that was a huge, huge breakthrough in the field, understanding this modular basis of gene construction. And for our purposes right now, what the most interesting consequence of that is it's no longer one gene, one protein. One gene instead can generate all sorts of different types of proteins, different settings, different circumstances.